All right, here we go. The difference between protein synthesis and DNA replication. So let's start what we already know about, which is DNA replication, which we've talked about in previous chapters. And then right now we're focusing on protein synthesis. All right, tell me by definition what is the beginning and the end of DNA replication? What is the beginning and the end of DNA replication? We start with what with DNA replication? The strand of DNA, right? And we get what? Two strands of DNA, right? You like my
artistic ability?
Isn't that great? What is that? What is also special about these strands of DNA? They're the same. They're identical. Identical.
Identical.
I knew that looked wrong as soon as I said it. Now the whole world can see how bad a speller I am. One strand, two strands. What are some key players? Like, what are some proteins or some enzymes, which are the same thing, enzymes are proteins, that we would use DNA polymerase, okay? So we need some DNA polymerase, right? And what's the function of DNA polymerase? Unwind it, which is cool that we say unwind because you guys read unwind, really? So that's how it goes together. No, it unwinds the DNA, and then what does it add to each side? RNA molecules? Nucleotides, right? Nucleotides. Remember, within the nucleotides, we have these pentose sugars, we have the bases, which are very important in the whole code, and then we have the phosphate group. So basically, these guys are just being added. So, what do we get in the beginning? One strand of DNA, two strands of DNA, identical. That's DNA replication. Does that make sense?
Yes.
Okay, awesome. Protein synthesis, holy cow, how much different can you get, right? We start with, again, do we start with DNA?
Yes.
And what do we end up with? Specifically, what type of protein? No? Polypeptide, right? Which is basically the beginning of a protein. Okay? Is this making sense? Now, how does this all take place? Well, it takes place several places. Over here, this all happens in the nucleus, right? Where does this happen over here? Wow, it starts in the nucleus, right? But it ends up in the ribosome, isn't that correct? Yes. So there's two sites that are important. What else is important here? What else is important? Who are some players here? Instead of DNA polymerase, we have RNA. Ah, yes. That might have been loud enough that actually my iPad actually picked it up. Yay. RNA polymerase is going to... Now, are these polymerases essentially very similar? Are they both enzymes? Yes. Are they both adding nucleotides? Yes. Are they both unwinding? Yes. Yes, but not teenagers. So they're unwinding the DNA. And the problem is here, let's get some colors in here. So if I have DNA and I unwind it here, okay, and I have, well, this is DNA, so there's no U's, right? If I have these 6 bases right here, then mRNA is going to come and copy it, right? But does it copy? It transcribes it. So this is transcription, right? So this is transcription. So instead of A, this would be U. No, it'd be A, U, G. It's kind of a funny G, A, U, C. Of course, it's going to be longer than this, but we're just going to simplify it. Then what does the mRNA do? leaves the nucleus, goes to the ribosome. It's going to lay down on the ribosome. And then we know with this AUG, AUC, this is going to provide the directions for what to happen. Well, now we're kind of back to the whole codon, anticodon, which we just went over, right? And this is starting to make sense because then we have codons. We have codons right here. We have anticodons matching up with it, right? And it's carrying amino acids, which eventually we're going to get a bunch of amino acids, and we're going to have a polypeptide. Does that make sense?
Yes.
Okay. All right. Okay, well, set this baby off here.